Finding the cause of human genetic disease is a really complex area of research. Thousands of people across the world work on it. These are the diseases like cancer and heart disease that affect millions of people in the world. And because so many people get these diseases, there are also very many different genetic causes for these diseases, and the complexity just makes it really hard to find the disease genes. Dogs get all the same diseases as we do. They get, cancer, heart disease, epilepsy, all those same kind of things. They have roughly the same genes as we do. They go to doctors or veterinary medicine, and they have the same environment as we do to a large degree. So they're very similar.
With LUPA, we try to understand the genetic component of complex disease using dogs, because dogs are suffering from the same disease that we are suffering from, but the genetic is less difficult to understand.
English Springer Spaniels get breast cancer, and whereas that's not necessarily a good thing for the dogs, we can now use this to understand these diseases and then help both dogs and humans in the process. What we do is that we take 100 dogs like Stella who have breast cancer in the English Springer Spaniel breed, and then we take an equal number of healthy dogs, and then we compare their entire genomes, and we find the region where all the sick dogs' genome look special. And then we know that the disease gene is there.
They looked to the nose and to the lungs, and they took examples of it, and also X-rays, and they saw that the heart, for example, and other stuff was on the other side of the dog, what not was normal, but was normal for a PCD.
And so what we hope is that the gene that will be identified will also be responsible for some of the human PCD cases, but not recognized as such so far. That is not discovered in the human, because it's more difficult to study PCD in human. So what you see here on the right is a cross-section through the cilium or acilium of a healthy dog. And so these dark doublets which you see, you can consider that as being the skeleton of the cilium. And that's the way it should look. And when you have it like that, it functions properly and it has the right shape. But in these affected Old English sheepdogs, as you can guess here, the structure is abnormal. You don't have this nice circle. All things seems to be disorganized.
The particular aim here is to help both man and his best friend. We can do that by developing genetic tests to the genes we find, both in dogs and in humans. And we can also, in the long run, really find new drug targets and therefore find much better treatment options for the diseases, hopefully both in dogs and in humans.
So it's exciting when you know that people are working on those diseases for a very, very long time, and now we are able with our best friend to find the genes responsible for those diseases. Yes, it's very nice to work within LUPA.